Welcome. In this lecture, microscope and its importance in microbiology lab. The microscope is an essential tool in microbiology lab. Microorganisms such as bacteria, fungi, viruses are too small to be seen with the naked eye. Microscope allows scientists and lab technologists to magnify these organisms so that they can be studied in detail. Two main types of microscope used in microbiology lab, and there are many other microscopes we will speak about: the light microscope and the electron microscope. These are the most common used microscope in bacteriology lab. Light microscope use visible light to illuminate the specimen. They can magnify object up to about 1000 times. Electron microscope use a beam of electron to illuminate the specimen. They can magnify object up to millions of times. Light microscope are the most common type of microscope used in microbiology lab. They are relatively inexpensive and easy to use. They can't magnify an object as much as electron microscope. Electron microscope are more expensive and complex to use, but they can provide much more detailed image of microorganisms. It gives us the microorganisms size and shape, the cell structure, the motility, movement, arrangement of cells inside the culture, presence of spores or capsules. By studying microorganisms under a microscope, microbiologist or lab technologist or technician can gain a better understanding of their biology and how they cause diseases. Also, this information can be used to develop new diagnostic test, treatment, and find historical background about the microscope. Invented shortly before 1600, either by Zacharias, Jensen, or Galileo. This two person also in development of the microscope. There are two persons in the 17th century, Hooke and Leeuwenhoek. Hooke in 1665 discovered compound microscope. He was able to see individual cell of a living creature. Leeuwenhoek in 1773 with his single lens microscope with magnification power 300x discovered the microbial world. He described the raw microorganisms for the first time. He published his discovery in Journal of Royal Society of London in 1683. The early microscope was called the simple microscope, consists of biconvex lens and were essentially magnifying glasses. Was called the simple microscope, consists of biconvex lens and were essentially magnifying glasses. To see microbe, a compound microscope which has two lenses between the eye and the object. This system magnifies the object illumination of the system. This is the consistency of the microscope. The illumination system, either the sun, the mirror, the lamp, a source of light, ensure adequate light is available for viewing. The bright, also the light microscope, they called bright field microscope, because in the illumination by the light to differentiate it from the dark field microscope. Bright field microscope, first to be used in lab, allow light to pass directly through the eye without being deflected by an intervening object in the of the microscope, the base and the light source, the diaphragm, also stage. This is a stage, diaphragm, stage, the condenser, the object, the revolving nose piece, stage, arm, support the body. This is the eyepiece, the coarse adjustment knob, fine adjustment knob, junction joint, the condenser, the objective length, revolving nose piece. The basic frame of microscope consists of the base, the stage to hold the slide, an arm for carrying the stage, may have two clips or movable mechanical stage to hold the slide, and the light source in base. Condenser consisted of several lenses which concentrate light on side by focusing into a cone as iris diaphragm.